Good evening, everyone. On behalf of Zydus, I, Dr. Suyash Bharat, welcome everyone. We are very much looking forward for today's session altogether. We would like to thank MSG and Dr. M.B. Agrawal sir for this wonderful opportunity. And with this, I'd like to hand over session to Dr. Amit Khurana sir. Over to you, sir.
Thank you, Dr. Suyash. Well, very good evening to all of you, and good morning and good afternoon to all of those who are joining from rest part of the world. Today, our guest speaker for the day is Dr. David Tichy from Philadelphia, USA, and he's going to lecture us on childhood ALL further advances. This webinar is brought to you by Mumbai Hematology Group, supported by Zydus, dedicated to life, and very well managed by Perfect Square. I thank Mr. Sumit Madan, Mr. Ashish Kumar, Mr. Rajesh More, and team from Zydus, Mr. Yash, Mr. Kalpesh, and the team from Perfect Square, Executive Committee of Mumbai Hematology Group. Our guest speaker for the day, Dr. David Tichy from Philadelphia, USA. All our discussants were themselves very eminent pediatric hematologists, and you participants for sparing your Saturday evening, morning, or afternoon from whichever part of the world you're logged in. Tomorrow, that is Sunday, 15th of February 2026, from 11:30 a.m. Indian Standard Time and 6 a.m. UK time, we have Dr. Shankara Panisha from Birmingham, UK, speaking to us on Zenubrutinib, the new kid on the block. Let me introduce our discussions for the day, and we have a galaxy of them here displayed in alphabetical order. I will pause for a while for you to look on all our discussions for the day for very eminent hematologists, hemato-oncologists, medical oncologists, as well as pediatric hematologists all across the country. Some more over here. So these are our discussions for the day. And now it's time to introduce our guest speaker for the day. We have with us Dr. David Tichy from Philadelphia, USA. Dr. David Tichy is Professor of Pediatrics and Endowed Chair in the Division of Hematology and Oncology at Children's Hospital, Philadelphia, Perelman School of Medicine at University of Pennsylvania. He is the chair of the ALL Disease Committee for the Children's Oncology Group. His areas of expertise include ALL and immune dysregulation disorders, including ALPS. He did his medical school from Eastern Virginia Medical School, Norfolk, Virginia. His pediatric residency was at Children's National Medical Center at Washington, D.C. His fellowship in pediatric hematology and oncology was from the Children's Hospital of Philadelphia. He was postdoctoral research fellow in pediatric hematology and oncology at the Children's Hospital of Philadelphia. Today, he's going to lecture us on childhood ALL and further advances. Well, thank you so much. I stop over here. Over to Dr. David Tichy.
Thank you so much for the kind introduction. Let me share my slides real quick. Can you see my slides okay?
Perfect, sir. Please go ahead.
All right, so I'm gonna get thanks. So thanks again for inviting me to talk today. I'm going to talk about childhood acute lymphoblastic leukemia and further advances. These are my disclosures. I won't go through them. The improvement in cure rates for patients with acute lymphoblastic leukemia, I think, is one of the great success stories in modern medicine. Previously incurable disease, and nowadays in 2026, the majority of children are cured, but the rates of cure vary across the globe and are higher in some parts of the world than others. And we still have a lot of work to do because the goal is to ultimately cure every child with acute lymphoblastic leukemia. Historically speaking, patients with T-cell acute lymphoblastic leukemia did worse than those with B-ALL. ALL has two flavors, B-cell and T-cell acute lymphoblastic leukemia. However, with modern therapy, their outcomes are very similar. And how did we get here? How did we get to the point where we can cure most children with acute lymphoblastic leukemia? Most of it is for using drugs that were approved in the US between the 1950s and early 1980s. Most of the rest of the world within the same kind of time frame as well. And we really spent the past 30, 40 years studying these same drugs in different combinations and different schedules, kind of rearranging the deck chairs to get to the point where we are today. Even though we've improved cure rates in newly diagnosed children through the use of cytotoxic chemotherapy primarily, although more recently with immunotherapy, which I'll get to at the end of the talk, there's still some areas where we have huge needs for improvement, one of which is relapse. While the outcomes for children with newly diagnosed leukemia have gotten better, those of relapse leukemia remain poor. They're getting better in B-ALL, probably mostly driven by immunotherapies, but relapse T-ALL and infant ALL in particular have pretty dismal outcomes that haven't changed that much in the past 20, 30, 40 years. There are other places that we still need to do better in the front line as well. Hypodiploid ALL is just one example of that as a part of bad biology that continues to do poorly in a chemotherapy era. We'll have to see if immunotherapy changes that. Infant ALL is another area where historically we had done poorly, and intensifying chemotherapy using things like bone marrow transplant haven't made an impact. However, the use of immunotherapy is actually finally starting to make an improvement in cure rates, we think, in infants with ALL, at least based on some data that are coming out of Europe that I'll talk about at the tail end of the talk. And one thing that's been historically done over the past 30, 40 years, really to improve cure rates, is intensifying chemotherapy for patients with poor risk or more likely or less likely to be cured, and keeping therapy relatively modest or even trying to reduce therapy in patients with favorable risk disease. In the Children's Oncology Group in the early aughts, so about 20 years ago, we ran a number of clinical trials where we continued to try to intensify therapy as a way of improving cure rates. I'm not going to go through these trials, just to say that all of these trials were unsuccessful in improving cure rates even more just by giving more chemotherapy. So we've hit a point where just giving more is not going to be the solution to improving cure rates. So if that's the case, then how do we really focus on improving cure rates moving forward? And I'm going to focus on 4 different areas over the next hour that people are looking at for improving outcomes in patients with ALL, and those including improving risk stratification, introducing new targeted immunotherapies, and then trying to understand disadvantages in historically disadvantaged populations to mitigate healthcare inequities as a way of improving outcomes. So I'll start by talking about things that are going on to improve risk stratification in ALL. So risk stratification is basically means integrating clinical variables, demographic variables, somatic tumor biology, and clinical response to put patients in different risk groups and tailor therapy accordingly. Different cooperative groups in different places in the world use different thresholds and cutoffs for some of these things, but most of the factors in BLL are pretty consistent. Most cooperative groups risk stratify patients based on age and presenting white count. Most include CNS status, or how much disease you have in the CNS space when you present. Most cooperative groups use MRD, and that can be a combination of blood or marrow MRD at different time points. And then most cooperative groups also use somatic tumor biology. Some groups use different genomic alterations than others, but essentially for combining all these different factors, you can put patients into different risk groups with different cure rates. For example, in BALL, the Children's Oncology Group of our current risk groups, their outcomes range from greater than 95% to down, and this is just standard risk ALL being shown here in some of the high-risk patients, to groups that have less than 65% survival. One thing that's changed over time is risk stratification is getting far more complex. It used to be that we would primarily, from a genetic standpoint, focus on primary leukemic drivers like ETV6-RUNX1 or hypodiploid ALL, BCR-ABL ALL. However, nowadays we realize that there's cooperating lesions that also can impact outcome, and try to figure out better ways of using those cooperative reasons and multivariable models to actually predict outcomes better. On the left is some data that were presented through the Target Initiative by Brady and colleagues a few years back, that they actually looked at cooperating mutations. They did look at a lot of things in here to see if those impacted outcomes. And without going through the data, for example, patients with ETV6-RUNX1, which we think is a favorable subtype, there were patients who had alterations in a gene called TBLXR1 that had inferior outcome compared to those who didn't. And this was just a similar theme, that for a lot of different genomic subtypes, there were secondary or tertiary lesions that could also impact outcomes. A paper that was published by Mignon Lo and others through an initiative that was funded by the NCI called the MP2PRT showed and also looked at different types of genomic alterations in favorable risk kids to see who are the patients that we think are going to do really well that relapse. And one thing that they identified was that something like DV6-RUNX1, if it was a balanced versus an unbalanced translocation, that actually significantly impacted outcomes, for patients with unbalanced translocations did not actually have the superior outcomes we normally think of with ETV6-RUNX1. In our next generation of trials that are open in the Children's Oncology Group, for example, we are going to take this into account of the type of translocation that's involved. Another thing that's been noticed by a number of groups across the globe is for some genetic alterations, it's not just which gene, it's how it's altered that can actually impact outcomes. So on the left here is looking from that same MP2PRT paper that looked at different types of PAX5 alterations. And without going through the different types, as you can see, there were some patients with no PAX5 alteration that had really good outcomes, but depending on the way that PAX5 was altered, it actually did significantly impact outcomes. That's on the left, and on the right is looking at IKZF1 alterations, where patients without a IKZF1 alteration do the best. Patients with IKZF1 alterations do worse in general. There are exceptions to that, and there are combinations of IKZF1 of other alterations, something called IKZF1 plus, defined differently by different cooperative groups, where they have cooperating mutations in things like CDK N2A or ERG that could change your risk with that. And with one particular definition of IKZF1 plus, those patients did markedly worse than those who just had IKZF1 alterations, just to highlight that risk stratification is getting more complex. And I think we're moving into an era of probably using things like AI to help risk stratify patients. A lot of the variables we use, we treat them as their dichotomous variables. For example, age over or less than 10 determines if you're NCI standard risk or high risk. Same for white count greater or less than 50. But we know that it's not just a pure cutoff. If your white count when you walk in the door is 51 versus 49, that really shouldn't make such a huge difference in outcome. And it's probably more of a continuous variable. And we're starting to think about that in risk stratification modeling. And I think because of the complexity of genomics, I think one thing that's starting to happen across the globe is trying to move towards more modern genomic technologies. There was a nice paper that came out of Europe last year that actually compared the cost and turnaround time on using more modern things like SNP arrays and RNA-seq versus more traditional ways of risk stratifying, including routine cytogenetics, FISH, and other things. And because of the number of tests it is required with typical genomic risk stratification versus with more modern, where you use more expensive technologies like RNA-seq, but you're able to get much more information off a single test, it's actually cost effective once you set up the technology to do it by technologies like RNA-seq. And another change that we're making in the Children's Oncology Group with our next generation of trials is we're going to start using RNA-seq and SNP arrays to risk stratify patients in an effort to try to move away from things like FISH and cytogenetics. TALL, which is an area that I've particularly been interested over the years, is even more complicated, or because we actually aren't very good at risk stratifying patients. And part of that's because the prognostic clinical variables that we use in BALL are not very prognostic in TALL. So without going into too much detail, in BALL, age is highly prognostic. If you're less than 1 or as you get older, you start to do worse. We've looked at this in TALL, and just we found that, for example, patients who are adolescents or young adults with TALL actually have equivalent outcomes to those who are younger, including younger children. White counts in TALL don't have the same prognostic significance. A white count at 50,000 is not prognostically different if you're over it than under it. There's some data that suggests very high white counts, like those over 200,000, may show inferior outcome, but even that has an interaction. Some data that came out of COG showed that a very high white count was prognostic in certain TALL immunophenotypes, but not others. And then there's differences in the literature as far as the prognostic significance of CNS status in TALL. And BALL, having no CNS disease that you can detect a diagnosis, what we call it CNS1, is great. Having CNS2 disease, which is low-level disease, does worse. And patients of CNS3 disease who have higher levels of CNS disease at presentation do much worse. In TALL, CNS3 does markedly worse. But in a lot of cooperative groups and a lot of studies have shown patients with low-level disease like CNS2 actually have the same outcomes as those with CNS1. So just to show differences between BALL and TALL. Indeed, in TALL, historically, the only prognostic factor that has been shown to be reproducible across groups is MRD. And in TALL, unlike BALL, it's really later time point MRD that's the most prognostic. So after 3 months of therapy, if you still have persistent MRD, those patients do really poorly. Early time point MRD, like at the end of a month of treatment, actually does identify a favorable risk group, but this is the majority of patients actually. So the majority of children with TALL relapse are classified as low risk at diagnosis because they're MRD at all time points, and we don't use other factors in risk stratification. I think that is finally starting to change in 2026 based on some genomic papers that have come out of my lab and other labs that have looked at the genomic landscape of TALL on a more broader scale to enable us to risk stratify. I'm just going to show a little bit of the data from a large project that's been run in collaboration with my lab and Charles Mulligan's lab at St. Jude's. And for this, we did whole genome sequencing, whole exome sequencing, and RNA-seq in over 1,300 patients of TLL treated uniformly on the ALL 0434 clinical trial, which I'll talk about later. We're now validating those results. We just finished sequencing the successor trial of that, ALL 1231. And through this large initiative, we're able to now divide patients of TLL into 15 different genomic groups that have distinct genomic drivers, gene expression patterns, developmental states, and outcomes. Some of these groups are novel, and we propose a new nomenclature. And I'm going to go through this briefly. One thing we were able to do for this large amount of work is actually identify the leukemic driver in the vast majority of cases of TALL. We were able to identify it in over 95% of the cases. Most of these drivers were things that we knew were involved in TLL before, things like Tau1, LMO2, et cetera. We just understand the frequency of these better now. We were also able to characterize the overarching genomic landscape of TLL as far as just genomic alterations that could be copy number alterations or point mutations or deletions. The most commonly altered gene in TLL is CDKN2A, followed by Notch1. Through this large work in a lot of patients, most of the genes that we found were ones that we knew about before. There were about 13 novel genes that we identified that were currently altered, but these were pretty rare, only affecting 1 to 2% of patients. So again, similar to what we saw with leukemic drivers with these other lesions, we understand the frequency and the type of alterations better through this large work. What we were surprised to find as far as the breadth and the depth of this was how important non-coding alterations are in TALL. Alterations in non-coding or intergenic areas of the genome. And this probably explains why we've not been able to really identify prognostic genomic alterations in TLL. These are things that we missed by and large with traditional exon-based sequencing approaches, including next generation sequencing. And also, unlike BALL, we can neatly put patients into a few big groups based on usually recurrent fusion oncoproteins. In TLL, we don't have that luxury because the biology is far more complex. For example, one mechanism of oncogene activation or oncogenesis or leukemogenesis in TLL was TCR enhancer hijacking, which basically means that another gene's enhancer turns on the T cell receptor, and that's what leads to leukemia. And we found this type of mechanism of oncogene activation occurred with over 40 different genes at different frequencies. So for example, LMO2 enhancing the TCR occurred in 82 out of 1,300 cases. We saw lots of different mechanisms of leukemogenesis, highly complex, lots of different partners. But again, the long and short of it was the majority of these were non-coding or intergenic alterations, things missed under typical exon-based approaches. As I mentioned, we were able to divide patients into 15 different genomic subtypes. This was by integrating their expression data with their whole genome sequencing. We then also did single cell sequencing of healthy normal thymus and bone marrow. And then we took the expression data we had from our TALL and we overlaid that into our normal single cell expression data to actually try to figure out where the cell of origin, or at least what is the developmental state the T cell blast most mirrored. And we found it did vary significantly based on our leukemic subtypes, with some leukemic subtypes being more stem cell-like and some being more like mature TALL. And we used a lot of this in our nomenclature. For example, just one example, there was this cluster which we called the LMO2 gamma delta subtype, is a new subtype of TLL. And the reason we named it that, it was a group of TLL with LMO2 alterations was the primary leukemic driver. And then on a normal developmental map, these TLL cells were most like gamma delta T cells. So we named it after that. For example, another group was the Tau1 alpha beta like group, and they had Tau1 and other alterations. But they're from us, the T cells mapped most like double or alpha beta T cells. There were a number of these different genomic groups that were defined based on a common deregulation of an oncogene. For example, this cluster here is the TXL3 group, and that was mostly made up by dysregulation of the TXL3 oncogene, most commonly by BCL11B enhancer hijacking, but we also identified a number of different ways it could be rearranged as well in TALL. And one thing I pretty should have said is with this expression map that I'm showing, each dot represents a case. The closer two cases are together by expression is how related they are, and that's how you can cluster these into different groups. One thing we also wanted to look at was the immunophenotype of the different genomic subtypes and actually how that influenced biology in the way that we think about it and possibly also outcomes. So TALL has been broken down into different immunophenotypes, the biggest one being the early T cell precursor immunophenotype, first described by Dario Campana in the early aughts, that has a unique immunophenotype I won't go through. Early papers suggested ETP immunophenotype patients did worse. However, more recent data says with modern therapy, they do just as well. However, there's been some mix in the data with some groups saying ETP patients do worse and some do better. We wanted to understand that better. So the first thing we did was we looked at patients with ETP by immunophenotype to see what genomics they had. And we found patients of ET immunophenotype fell into three different groups: the TXL3 group, the BCL11B group, and this new group, which we termed ETP-like, and I'll explain that. There's another immunophenotype called the near ETP immunophenotype, first described by Brent Wood, which basically meets the same criteria as the ETP immunophenotype. However, it's not meeting a CD5 negative or dim criteria. We also found that these cases clustered within the TXL3 and that ETP-like group, but instead of BCL11B, a lot of them clustered in the tau1 alpha beta-like group. So we really wanted to look and drill down on an immunophenotypic versus genomic-based classification to say which one is probably the more accurate and better one to move forward with in TLL. And I think it's really going to be the genomics, and I think the ETP-like group really highlights that. So if you look at our entire cohort of 1,300 cases in this cluster that we termed ETP-like, 70% of the ETP immunophenotype and 41% of the near ETP immunophenotype cases overall fell in this ETP-like group. If you just looked in that ETP-like group, it was basically made up of about a third of ETP, a third of near ETP, and a third of non-ETP immunophenotype. And really, it's the ETP-like group that does worse overall. This is showing a Kaplan-Meier curve of those 15 different genomic groups that I mentioned, where ETP-like is that purple line in the middle with inferior outcome. If you look within the ETP-like group and then compare those of the different immunophenotypes, what you find is they have similar genomics, similar genomic alterations, similar rates of induction failure, similar poor or high MRD at the end of induction, and similar poor outcome. So regardless of immunophenotype, the clinical and biologic features of patients within this ET-like group are the same. And if you look at outcomes within the ETP-like group based on immunophenotype, they're superimposable. I think a better way of actually looking at this and really highlighting the importance of genomics versus immunophenotype and TALL is this figure right here. Going left to right, three different Kaplan-Meier curves. The first is all the patients of ETP immunophenotype. The middle is those of near ETP immunophenotype. And on the right is those with non-ETP immunophenotype. And then breaking it down in those groups by those with the ETP-like genomic group versus everything else. And as you can see, regardless of immunophenotype, the patients who do poorly are those with the ETP-like genomic signature. So patients of ETP immunophenotype that are in other genomic groups, so example, BCL11B and the TXL3 actually have excellent outcomes. It's just the ones that have this ETP-like genomic group, which totally supersedes immunophenotype and really explains why we see these differences in outcome across groups. It's because probably different groups, the percentage of patients in the ETP-like genomic group, as far as their immunophenotype, probably varies. As I did mention, though, for the first time, we're now able to kind of predict patients' outcomes of TLL based on their genomic risk group, based on the sample size of this and the inclusion of whole genome sequencing. As you can see, some groups do really poorly. Fortunately, they're rare. So two of the low lines here are the LM2 gamma delta like group that I mentioned before, and this other group called SPY1 rearranged. Fortunately, each of these were less than 20 cases. One thing that we also found in TLL, which makes the risk stratification even more complicated, is it's not just necessarily which gene is altered, it's how it's altered. So people usually think of notch alterations in TLL as being favorable. And what we found was that most notch alterations are favorable in TLL, but there's this group of patients who have Notch1 intragenic loss. So they have non-coding alterations in Notch1, and those patients actually have inferior outcome. And this is not the only gene that we saw this. There were a lot of genes that we saw that it wasn't just, is the gene altered, yes or no? It's how it's altered actually has a strong impact on survival and outcome, which just makes it more complicated as we're developing risk models. All that said, we are able to start developing some multivariate models for better risk stratification of TLL. This is one model that we developed, which incorporates RNA-seq genomic sequencing data and MRD to put patients in 8 different risk groups with event-free survival ranging from 45 to 98%. Maybe a group that, for example, really high outcomes, this 98% group, where we could start talking about turning down therapy or de-intensifying care of therapy, versus patients who have 40 to 50% survival. We know chemotherapy alone isn't going to cure them, and those patients probably might benefit from a transplant in first remission. We were also to identify a number of genomic signatures or expression signatures that could actually be used to correlate with outcome. This is just showing one of them. This was actually a signature which we'd call a bone marrow precursor-like signature. We didn't actually identify it in the bulk sequencing. We had another project where we did single-cell omics on a cohort of cases where we compared single-cell profiling in patients who later had refractory disease versus patients who did very well, identified a high-risk signature, and then applied it to the overall genomic cohort, finding it was highly prognostic. A number of other groups have also recently published, a lot of it using the data that we published, that large sequencing project, identifying other genomic signatures that are correlated of worse outcome that I think could be translated to the clinic. It does require its expression data, though, so it does require an RNA-seq typically to actually be able to risk stratify using these kind of models. And the last thing I will mention in risk stratification, which I think is new and pretty exciting, first published by Steinicki and colleagues, and we have some unpublished data in this area as well, that's using methylation to subdivide both B-ALL and T-ALL and AML into different biologic groups. And it turns out that most of our major subtypes of leukemia have unique methylation signatures, and it's far easier to do methylation-based risk stratification in the clinic because it can be done it's done through DNA sequencing using something like the EPIC chip, for example, which is very reproducible and relatively cheap. We actually looked and did methylation, which these are unpublished data, on over 400 cases that I talked about with the big genomic sequencing data, and we're now validating this in another 400 cases on a different trial. But as you can see here, this is methylation signature. We can identify those 15 different groups by unique methylation signatures. So this might be an easier way of trying to move these genomic data into the clinic. I'm now going to shift gears a little bit and talk about some targeted therapies and immunotherapies that we're using in ALL. As everyone on this call knows, BCR-ABL1 really changed the paradigm here. It was really, we could argue, the first type targeted therapy that was used successfully to treat a malignancy, i.e. not a cytotoxic chemotherapy. Earlier data, which included used tyrosine kinase inhibitors like imatinib or dasatinib, show that those take BCR-ABL ALL and actually make it much easier to treat and far better cure rates than chemotherapy alone. One subtype of ALL that's had particular interest in the B-ALL space has been PH-like ALL. This is a subtype of B-ALL that has a gene expression profile that looks like BCR-ABL1 ALL, but or BCR-ABL1 negative. It's complex biology because these cases can have dysregulation of different genes that are involved in JAK-STAT signaling, or they can have ABL class alterations that are not BCR-ABL, or they can have a whole bunch of other alterations, including things like dysregulation of MAP kinase signaling. There's been a number of clinical trials that have looked at using TKIs in patients of ABL class alterations, and then looking at JAK-STAT inhibitors of patients of JAK-STAT alterations, including CRLF rearrangements. We're waiting on the data for some of the JAK-STAT inhibitor studies. Some of the early stuff that's come out with the ABL class alterations in PH-like suggests that TKIs may improve outcome, and they're now becoming more standard of care in patients of PH-like ALL with ABL class fusions. Tai Tran and others published a nice How I Treat paper in Blood in 2025, just last year, talking about when they're intervening with targeted therapies in patients with PH-like ALL. And essentially, those with ABL class alterations, they're doing it as soon as they identify those and treating them with chemoimmunotherapy. And I'll get to that later in the immunotherapy part of the talk. Versus patients of JAK-STAT alterations where the data are more thin and we don't actually know if adding a JAK-STAT inhibitor helps, they're reserving that for patients with highly refractory disease. Another targeted therapy, and I'm not going to go through all the targeted therapies for B or TLL because there are just too many. I'm just going to highlight a few examples, is menin inhibition for KMT2 rearranged ALL. As people know, KMT2 rearranged ALL is a subtype that tends to do worse, and that's regardless if it's B-ALL or T-ALL, and they do particularly poorly if they're infants with ALL. Menin inhibitors are a way of getting at the KMT2 the MLL fusion product by binding to the pocket of menin, which is important for turning this on and leading to differentiation through Hox genes. And there's been a number of studies that have looked at adding menin inhibitors in early phase trials in patients with KMT rearranged ALL. And there's also a number of trials right now looking to see if they'll actually improve outcomes. Just as one example, there's a trial for the Children's Oncology Group, the ALL 2121 trial that's being chaired by Kelly Falk, that's adding a menin inhibitor into either an AML-like backbone or an ALL-like backbone for infants with KMT2 rearranged ALL. That study is actively accruing, and we anxiously await its results when it completes. In addition to targeted therapies for B-ALL, there's a number of targeted therapies that are being studied right now in the clinic in patients of T-ALL. I'll just show a couple quick examples. We have moved some novel therapies into the clinic with patients with TLL, things beyond just traditional cytotoxics. One was Nelarabine that was introduced in the ALL 0434 clinical trial. This is a trial that was chaired by Stuart Winter and Kim Dunsmore. It included a 2x2 pseudofactorial randomization, randomizing patients to receive or not receive Nelarabine, or to receive or not receive, or between two different types of methotrexate, either Capizzi methotrexate or high-dose methotrexate. Capizzi methotrexate was shown to be superior to high-dose methotrexate, and patients who received Nelarabine had improved disease-free survival with TALL, but not those that were also included in the randomization of T-cell lymphoblastic lymphoma did not seem to benefit from Nelarabine, probably because most of the benefit in Nelarabine was reduction of CNS relapses, which isn't an issue in T-cell lymphoma. The successor trial to that was the 1231 trial, and this was the trial that we added the proteasome inhibitor bortezomib in a randomized fashion and induction and delayed intensification in patients of TLL and T-cell lymphoblastic lymphoma. On this trial, we kind of saw the opposite results, where patients of TALL saw no benefit from bortezomib, but patients of T-cell lymphoma had improved event-free and overall survival with the addition of bortezomib. I think it's important just to highlight that in T-cell disease, we've kind of harmonized therapy and treated T-cell ALL and T-cell lymphoblastic lymphoma the same when it came to using conventional cytotoxics. But now that we're moving novel therapies into the clinic, we're finding the TALL and T-cell lymphoma are not the exact same disease, and they're having differential response, and we need to understand the biology of these differences to know the best way to treat patients. Another thing that we did on the 1231 trial was, through some changes in the backbone, which I won't go through, we eliminated radiation in the majority of patients. On 0434, over 90% of patients received prophylactic radiation or therapeutic radiation. On 1231, it was less than 10% of patients. And without going through this, we found that by eliminating radiation through changes in the backbone, we had similar event-free and overall survival and rates of relapse. And mostly this was through intensifying the steroids and asparaginase a little bit as well. On our next clinical trial, which is opening in the next month or two in the Children's Oncology Group, we're not radiating any patients with TLL on that trial. They're all going to get treated with chemotherapy alone. And in the US right now, the standard therapy for children of TLL and T-cell lymphoblastic lymphoma, based on the results of this trial, is shown here. Patients with TLL were using a dexamethasone-based induction to get more CNS penetration with Nelarabine and a single IM of Capizzi methotrexate. T-cell lymphoma now we're treating different. We're giving them a prednisone-based induction because they don't benefit from the extra CNS penetration of dexamethasone. They just get the toxicity of that. And instead of Nelarabine, we're using bortezomib. Other novel therapies that are being looked at in the clinic, one of which is similar to PH-like ALL. In the ETP-like subtype of ALL, they have a tendency to have dysregulated JAK-STAT signaling pathways, regardless of their underlying immunophenotype. We had done some earlier work suggesting that ETP immunophenotype, and more specifically the ETP-like subtype, may benefit from JAK-STAT inhibition, and there are clinical trials looking at this. I think one thing we'll be able to use our genomics, maybe in the clinic now, to help guide therapy is we do know if those 15 different genomic subtypes, there are different frequencies of dysregulation of signaling pathways, some of which are targetable. This is just highlighting JAK-STAT alterations based on a subset of those genomic subtypes. And what you can see here is some subtypes like BCL11B, about 80% of cases have dysregulated JAK-STAT signaling. HOXA9 TLL, over almost 90%. Versus if you look at some subtypes like KMT2 rearranged, about 5% of cases have dysregulated JAK-STAT signaling. So if you had a patient with relapse disease and you knew they were KMT2 rearranged, giving them a JAK-STAT inhibitor would make no sense. Versus if you had a patient with BCL11B TLL, in theory, they may benefit from a JAK-STAT inhibitor. And sometimes for these highly refractory relapse patients, we're just trying to find some drug that may be effective, and we need more basket type trials to test these hypotheses. Another area that we're looking at studying in TALL is targeting Src kinase signaling, and this is based on data from Jean-Pierre Bouquin and Jun Yang, who did preclinical data, and they found that in a petri dish, at least, about 40% of cases of TALL are sensitive to the tyrosine kinase inhibitor dasatinib, and they found this was through dysregulated Src signaling. In particular, LCK is abnormally phosphorylated in about 40% of cases of TALL. It tends to be more mature TALL, and there's some trials that are ongoing right now looking at adding tyrosine kinase inhibitors into relapsed and refractory patients who are predicted to respond. Another area in TALL is targeting the apoptotic machinery. So over a decade ago, some groups showed that patients with more early lineage TALL tend to be addicted to BCL2 or dependent on it, and one more late lineage are BCL-XL dependent. Wendy Stock ran a clinical trial where they used a BCL2 inhibitor, venetoclax, and a BCL-XL inhibitor, navitoclax, in patients with TALL, and included 7 children, and found a very nice 71% CR rate in patients with relapsed and refractory disease. This has led to a number of trials looking at venetoclax and TALL. And there's been a lot of exciting data, I think, actually coming out of using different regimens, including venetoclax and azacitidine and TALL, showing pretty remarkable responses. And again, more likely if you're early lineage disease, but Ven/Aza is becoming one of my go-to regimens now for patients with refractory TALL or multiply relapsed when conventional drugs aren't working. My group actually looked at targeting BCL2 in that BMP-like and ETP-like groups that I mentioned before, and without going into the data, and this was in collaboration with Jun Yang, we found that cases of TALL with early lineage of these particular genomic signatures tend to be addicted to BCL2 and respond to BCL2 inhibitors, at least in a petri dish. And then the last pathway I'll mention very briefly as far as targeted therapies in TALL is targeting cyclin-dependent kinases. A number of groups, including Ioannis Iofannis, have shown that cyclin-dependent kinases are frequently dysregulated in TALL, including cyclin D3. There are drugs like CDK4/6 inhibitors that are used to target these pathways, approved in a lot of countries for breast cancer. In preclinical models, they were shown to be effective, and Elizabeth Raich ran a clinical trial that treated a small number of patients with highly refractory or relapsed disease, adding a CDK4/6 inhibitor, palbociclib, to conventional chemotherapy, and found some nice responses in some highly refractory patients, including like 5th relapse TALL. There are other ones that are under development as well that I won't go through now, but I think we're just at a nice era where we can take some of these genomic data and identify different targeted therapies that may be effective in some of these different subtypes of TALL, but we need clinical trials to kind of validate this. And the last thing I'm going to highlight from a therapeutic standpoint is kind of talking about immunotherapies and some of the newer work in that space. I think, as everyone knows, in B-ALL, immunotherapies have really transformed the therapeutic landscape and really changed the way that we're treating our patients across a lot of the globe. There's a lot of different types of immunotherapies. There's bispecific T-cell engagers like the blinatumomab. There's naked and conjugated monoclonal antibodies like inotuzumab, and there's also CAR T-cells. Blinatumomab was first shown to be effective in relapsed and refractory disease in studies that were run in the late 90s, early 2000s. I'm not going to go through the data. Some data in adults, followed by some data in children, show that blinatumomab seemed to be highly effective in the relapse setting. In COG, Patrick Bown was the study chair of the ALL 1331 clinical trial. This was a trial that in first relapse B-ALL randomized patients to receive a traditional chemotherapy backbone or chemotherapy adding in blinatumomab. And there was 2 risk groups on the study. There was those who were going to go to transplant who had higher risk disease, and there was those who were going to treat it with immunotherapy alone. And the long and short of it is the study closed early based on superior outcomes in patients treated with blinatumomab, both efficacy and reduced toxicity. Based on those compelling results in the Children's Oncology Group, we ran the ALL 1731 clinical trial. This is a trial that was chaired by Sumit Gupta and Rachel Rao that was just published this past year in New England Journal of Medicine. This trial was for NCI standard risk B-ALL and risk stratified patients into 3 groups. The most favorable group we call standard risk favorable, which is not shown here. They did not participate in the blinatumomab randomization. And then patients with average risk or of relapse or high risk of relapse of NCI standard risk were randomized to receive or not receive 2 cycles of blinatumomab on conventional chemotherapy. The backbone was different in the average and the high risk patients. It's more intensive in the high risk patients, but blinatumomab was added in the same section, the same places, and it was 2 cycles. These results were just published, as I mentioned. This study was closed very, very early at the second interim analysis, which was the earliest we ever had any trial on COG close, because there was such a difference in outcomes that was seen that early. These are looking at some of the 3-year data, and they're holding up at least at 4 years now, showing that patients who got blinatumomab plus chemotherapy had markedly improved outcomes, almost a 10% improvement in disease-free survival. And this was not only in the overall cohort, this was also held up if you broke patients into standard risk average or standard risk high. They both had a significant benefit from blinatumomab. We found this benefit was because these patients had less relapses, and we saw this overall and in the 2 different risk groups. And primarily we saw this was because it reduced, not surprisingly, bone marrow relapses. So blinatumomab doesn't cross the blood-brain barrier. We found that isolated bone marrow relapses went down remarkably, or combined relapses in patients with BLL treated with blinatumomab as compared to chemo alone. There was no difference in the rate of CNS relapses. Fortunately, there also wasn't a doughboy effect of some therapies. If you just reduce relapses in one compartment, you see a counterbalance of increased relapses in the other. That's what we see with radiation in TLL. If you radiate patients with TLL, you have less CNS relapses, but that's counterbalanced by more marrow relapses. It just shifts how you relapse, and your overall rate of relapse is the same, and your overall cure rates are the same. With blinatumomab, we found that it reduced bone marrow relapses, but that was not counterbalanced by increased CNS relapses. So your overall relapse rate was lower. And this is just one study showing a benefit of blinatumomab in the frontline area. As I mentioned before, there was a study that was done out of Europe from Princess Maxima, published in New England Journal of Medicine by Van der Sluis and colleagues, where they added a single cycle of blinatumomab to chemotherapy and then compared that to chemotherapy historical control. This was a small study, only including about 30 babies, but the difference in outcomes was really striking with, for the first time, actually getting, and these were early, like 2-year DFS data, but showing around 80 to 90% survival in infants with ALL. And these data are actually holding up with time. They presented some of the 4-year data on this trial more recently at a SIOP meeting, showing that it's, and ASH, that are showing that these results are consistent. So it looks like blinatumomab is changing the whole face of infant ALL, something that we haven't made any inroads in 50 years, really. Now we're, for the first time, able to cure more patients. As I mentioned, 1731 showed improvements in outcomes in NCI standard risk disease. The adult cooperative groups in the US ran the E1910 trial, where they added up to 4 cycles of blinatumomab to chemotherapy backbone, also showing markedly improvements of outcomes with blinatumomab. And based on results in infants, NCI standard risk, and adults with ALL, in the US at least, we took the leap and said, if it works in all those groups, why wouldn't it work in NCI high risk? And now pretty much the vast majority of patients in North America get blinatumomab as part of standard of care. The only groups at the front line of BLL that don't are those most favorable standard risk favorable group that I mentioned before. Although in our next trial, we're actually going to study blinatumomab in that population. It's not just blinatumomab that's been shown to improve outcomes or have some or nice efficacy in BALL. There's been data looking at inotuzumab, so a CD22 conjugated monoclonal antibody in relapsed ALL, shown in adults, followed by in kids, showing very nice efficacy. That has led to trials looking at inotuzumab in randomized setting. One study that we have ongoing in the Children's Oncology Group right now is the ALL 1732 clinical trial, which is a randomized trial, which is looking at the addition of 2 cycles of inotuzumab onto a chemoimmunotherapy backbone, where we're actually replacing some of our cytotoxic chemotherapy with inotuzumab on this trial. This trial was recently modified and redesigned and reopened to include blinatumomab on both the control and the experimental arm, and this just reopened to accrual in December. So it's going to take about 3 years to enroll in this trial, and hopefully we'll see inotuzumab showing a similar benefit and outcome as blina was shown to have in standard risk BLL. We have another clinical trial that's being shared by Aaron Breeze and Rishi Kocha, which is looking at in infants with ALL, both a targeted therapy and immunotherapy. So patients are non-randomly getting blina now, 2 cycles based on those those data from Europe, and this will be in a larger trial. So we'll see this will enroll far more infants with ALL. And then the randomized question that we're asking here is adding venetoclax into that. So it's adding both venetoclax and blinatumomab into a backbone of ALL. This study is open and actively accruing. It just finished a safety phase, and it'll be opening up to its expansion randomized phase in the next month or two. Titran is right now running an international trial, currently open for the Children's Oncology Group, but opening for the SFOL Cooperative Group throughout Europe for the IBFM and Altogether groups, hopefully next month. And this is looking at a non-random addition of blinatumomab and tyrosine kinase inhibitor to patients of Ph positive or Ph like BALL with Abl class fusions. For patients with Ph positive disease or Abl class fusions except for PDGFR beta, they're getting the TKI dasatinib. For those with PDGFR beta fusions, they're getting imatinib instead based on some preclinical data out of Europe. This is a single arm study. It was initially designed as a randomized study to receive or not receive blinatumomab, but based on the data that we saw that I mentioned before with blinatumomab, it seemed the incorrect. Nobody would randomize their patients to get blinatumomab anymore in Ph positive ALL based on the data out there. I think Ph positive disease is going to be the place that we're probably going to go to chemotherapy free or close to chemotherapy free the fastest, because we have the luxury of both being able to use targeted therapies with tyrosine kinase inhibitors and immunotherapies, really reducing or obviating the need for as much cytotoxic chemotherapy. And this study is really, it's a small pilot study really designed to understand what our event-free survival is going to be in a chemo immunotherapy tyrosine kinase era. So then we can design a randomized trial to test different approaches. And then the last immunotherapy trial that we have running through COG right now is the 1821 trial. This is being chaired by Stacy Cooper. This is the trial that's basically asking the question of, if we're giving blinatumomab to everybody in the front line, how well is it going to work in the relapse setting, and randomizing patients to receive or not receive a blinatumomab-based backbone in first relapse, or receive blinatumomab plus the checkpoint inhibitor nivolumab to see if that can overcome mechanisms of resistance to blinatumomab, which may be more important now in an era where many patients are getting blinatumomab in the front line. So another trial just opened this past year. Will take a few years to accrue, and we'll have to wait on those data. And then the final immunotherapy I'll just mention briefly is using CAR T cells, and I'm not going to go through a lot of the data here because it's a rapidly growing field. Some of the early CAR T cell work was looking at CD19 CARs. This is showing some of the early results of the Atlanta trial. It's an international trial using tisagenlecleucel or Kymriah in patients with relapsed and refractory disease, showing remarkable relapse-free survival rates. There's been a lot of CAR T cell trials that have been out there. This is showing one from Sloan Kettering, looking at more long-term outcomes of a CD19, CD28 zeta-based CAR, showing remarkable efficacy. There's also been CAR T cells in BLL that have targeted different antigens like CD22 that also look highly effective. They're now dual CARs targeting both CD19 and 22. I think as we're using more CAR Ts, it's getting interesting because we're seeing a lot of overlap as far as efficacy and toxicity, but a lot of differences as well. So, for example, CD22 CARs are more likely to for patients to get something called IECHS that they don't see very often of CD19 CARs, which is a secondary cytokine storm that patients get usually a few weeks after treatment that has abnormal macrophage activation, which we also see in cytokine release syndrome, but tends not to be as responsive to cytokine blockade as CRS. In TALL, immunotherapy is way behind for a number of reasons and hasn't really had the same inroads as BLL, but I think that's finally starting to change. There's a number of things that have made immunotherapy tougher in TLL. One is just the target and the toxicity of targeting it. If you have a CAR T cell that attacks normal B cells and malignant B cells, then you just give somebody IVIG to replace that. However, in TLL, if you wipe out your normal T cells and your T cell blast, you're basically replacing your leukemia for the equivalent of HIV. So it has to be a bridge to transplant unless we can identify antigens that aren't on healthy T cells. With CAR T cells, there's the issue of fratricide, which basically means if the antigen you're attacking is also on the CAR T cell itself, it would attack itself. T-ALL has also a lot of issues with just making a product when it comes to the CAR T cell world. There are host issues that their T cells aren't as healthy. It impairs manufacturing. These patients progress rapidly, so you can't often wait for a CAR T cell to be made or manufactured, and a patient can get too sick too fast. And then another issue is eliminating the blast from the products. So the last thing you want to do is transduce a T cell blast with a CAR and then give that back to a patient. So more regulatory things that make it tougher to make a product. That said, we're starting to move things in the clinic. There's been a number of groups that have looked at what you can target in T-ALL. I think a consistent thing seen across different studies have shown that things that are highly expressed pretty ubiquitously are things like CD5, CD7, CD38. So there's been a lot of interest there. My lab looked at, in preclinical models, studying the anti-CD38 monoclonal antibody, daratumumab, which we found was highly active in preclinical models. That led to the Delphinus trial that was chaired by Laura Hogan and Tina Batla, published in Blood in 2024, where daratumumab was given to first relapse T-ALL on a chemo backbone, which we were surprised and happy to see that about an 80% overall response rate after 2 cycles of therapy. And based on those results, there's a number of trials right now looking at the addition of daratumumab in patients of T-ALL. In COG, which I mentioned, we have a trial opening next month, hopefully, which is randomized patients on that standard chemotherapy backbone to receive or not receive 15 doses of daratumumab starting in consolidation with 2 strata for T-ALL and T-cell lymphoblastic lymphoma. This will be chaired by Eric Schaefer and Keith August. There's an adult trial, the EA9213 trial, that's being chaired by Shara Dinner and Tina Batla from the pediatric side, that's adding in daratumumab in patients with persistent MRD after a few months of therapy to see if that can get you to transplant. And then Mitch Cairo is running the AlloDart-T trial, which is treating patients with relapse disease who go to bone marrow transplant, who have resurgence of MRD or positive MRD going into transplant with daratumumab post-transplant to see if that can improve post-transplant outcomes. And then there's also been a number of studies looking at different CAR T cell products in patients of T-ALL. I'm going to just show 3 quick trials looking at different approaches to CAR T cells in T-ALL. The first came out of China. It was published in JCO in 2021. This was using a donor-derived CAR T cell, where basically they collected T cells from the transplant donor. And this was for patients who had a transplant. They went back to the donor and got T cells, or patients who are going to have a transplant. They collected T cells, made a CAR, treated patients with a CAR, and then bridged them to transplant with the same donor. A risky trial in that they could have had bad graft versus host disease. Fortunately, they didn't see that, but they saw a greater than 90% remission rate when they used this type of approach to treat T-ALL. There's a number of off-the-shelf CAR T cells that are being studied in T-ALL, while off-the-shelf hasn't been as attractive in B-ALL. In T-ALL, because of a lot of those issues of manufacturing I mentioned, it makes the concept of using a more of a universal allogeneic more attractive. And there's a 2-edited CAR from Wugen, a 3-edited CAR that was published from Great Ormond Street, and there's a 4-edited CAR from the Beam Therapeutic Group that my lab developed in combination that's currently being studied in a clinical trial. And the long and short of this is these off-the-shelf products are seeing very high response rates as well, over 70% across the different types of off-the-shelf CAR T. And then there's also been work doing more traditional CARs of auto CARs. This is work from Singapore by Dario Campana's group, published in Nature Med in 2024, where they made the problem with targeting CD7 is fratricide can be a major issue. So they made a CAR T cell that has a protein expression blocker, which prevents CD7 from being expressed on the surface of the T cells that you made the CAR out of. So you don't have that fratricide, and they saw an over 90% remission rate in patients treated with this CAR T cell. So I think an exciting time that we're seeing excellent response rates to donor-derived, off-the-shelf, and autologous CAR T cells. It just takes a little bit more work to build these things than we had in B-ALL, and hopefully it's going to start salvaging more patients of relapsed and refractory disease. And that's just some examples of it. There's many more that are in development. as well. And I'm going to skip, just based on time, because it looks like I'm getting toward the top of the hour, this section on healthcare and equities, just because I want to kind of talk about where the field is moving in the future for the last like 7 to 10 minutes before I finish. And all these things I've talked about so far are really focused on improving cure rates. But in an immunotherapy era, there's actually a lot of unique opportunities and challenges both in BALL and TLL as we think about where the field's going to move over the next few years. I think one question we have now is, now that we're seeing such an improved event-free and overall survival in patients with BAL in particular, can we start to de-intensify therapy? And if we're going to start to de-intensify therapy, the question is, what should we get rid of? As a physician, I can think of which drugs or which therapies I'd want to get rid of, but really what's causing the biggest impact on our patients, both short-term and long-term? And for this, Kelly Parker and Kim Buff, for the Children's Oncology Group, Kim Buff leading our patient advocacy group, ran a large survey through Mom Oncology asking patients and families what parts of therapy are the hardest for you, both what are the most difficult medicines and what are your biggest risks for the long term. And the most difficult medicine that patients identified were steroids, not too surprisingly. And then the long-term thing that patients and families are most concerned over, neurocognitive risks. So considering both of those things, our next trial for NCI standard risk BALL that we hope opens at the end of this year or early 2027 is asking some de-intensification questions and really focusing on those parts of therapy that patients and families are bothered by. So for standard risk favorable, for example, we're going to randomize patients to receive, who are not currently getting Blina, a blinatumomab-based backbone, but using that to eliminate delayed intensification and steroid pulses and vincristine pulses and maintenance. Versus our standard risk average group, we're going to keep in the Blina that we're using right now, but saying that the outcomes have gotten so good, we're going to use that to actually get rid of most of DI and also maintenance pulses. And then our higher risk group, we're actually going to ask both a de-intensification and a efficacy question by adding in a third cycle of Blina to get rid of a lot of chemotherapy as well. We also have a study in development, the ALL 2522 clinical trial. This will be chaired by Amanda Lee and Kieran Raven. The last trial I mentioned was being chaired by Ashley Pinchinette and Kathy Lee Miller. And this is trying to replace as much chemo as we can in patients with Down syndrome using a Blina and inotuzumab-based regimen to try to get rid of most of our cytotoxic chemo in most patients with high-risk Down syndrome. We also need to think about how we risk stratify our patients. I mentioned before all the work on risk stratification, but that's based on chemotherapy. Risk is really determined by factors associated with resistance to chemo. But we're now learning in immunotherapy era that some of our traditional high-risk genomic features may not be high risk in immunotherapy era. For example, pH-like ALL on the E1910 trial, if you look in the supplement, those with pH-like ALL who were treated with blinatumomab pretty much abrogated the poor outcomes in patients with pH-like ALL, so this may no longer be a unfavorable risk group in an immunotherapy era. On the flip side, there are probably going to be different genomic subgroups that do worse in immunotherapy era. So some data out of June Yang's lab looked at CD58 expression on different genomic subtypes of BALL, because CD58 is a ligand for CD2. It's important for T cell engagement, activation, and killing. And they had some data showing that low CD58 expression correlated with intrinsic resistance to blinatumomab because you can't engage those T cell engagers with a BiTE. And CD58 expression did vary based on your genomic subtypes. In particular, those of PAX5P80R had low CD58 expression, and those cases of BALL are intrinsically resistant at diagnosis to blinatumomab and less likely to benefit from it. So this could be as we are now using more and more immunotherapy and less chemotherapy, this subtype of BALL could become one that's becoming one of our highest risk subtypes because they're intrinsically resistant to Blina. We're going to have to start thinking about the changing landscape at relapse. Since blinatumomab and other immunotherapies are really impacting bone marrow relapses, but not CNS relapses, the percentage of cases that'll have CNS involvement at relapses will be much higher. So we're going to have to think about that as we're developing our relapse trials to make sure they're things that are the increased prevalence of isolated and combined CNS relapse in our relapse populations will impact trials. We need to think about who needs a transplant in first remission. Right now, similar to what I mentioned with biology and risk stratification, decisions on transplant in first remission are typically based on MRD or other features that lead to poor early event-free survival, so certain biology. But if immunotherapies have changed the biologies that are bad and also, in theory, are going to impact MRD rates, we need to retake a step back and say which patients actually need a bone marrow transplant. So on that ALL 1732 trial I mentioned in NCI high-risk BALL, we're no longer recommending that patients with persistent MRD at the end of consolidation get a transplant. We're actually recommending those patients get a block of Blina, reassess MRD, and if you still have persistent MRD after that, then they would go to transplant. But that could be a bad decision, but we're trying to frame shift when we're recommending transplant in those patients because we don't know when it'll be in a chemoimmunotherapy era. We really need to understand the late effects, both short and long term, of immunotherapies better. We assume that adding immunotherapies into our therapy are going to be better than traditional chemotherapy, but we don't know that. We do not know the consequences of wiping out healthy normal B cells in patients when they're children. We do know that patients who have common variable immunodeficiency, present when they're young, but they tend to have issues when they're older. And if you look at patients of CAVID, there's this cohort of patients who die in their early 50s of bad autoimmune disease and secondary malignancies. And what we need to understand is impacting our B cell numbers and function when you're young going to cause consequences down the road, similar to what we see in CVID patients. We have no clue on how immune therapies are affecting immunologic memory. For example, after treating somebody with a chemoimmunotherapy-based backbone, do they need to be revaccinated? If they need to be revaccinated, is it like a BMT patient where they need all their series again, or they just need a boost? And then immunotherapies cause toxicities. They can cause dysrhythmias. They can cause neurotoxicity in the form of ICANS. And we know that if you impact your heart and your brain with other medicines, even your neurotoxicity from sedation can cause long-term neurocognitive impacts. To think that immunotherapies that can damage our heart and our brains wouldn't do the same thing, I think is short-sighted. And then another thing that we're starting to think about now, but didn't even dawn on us before, is with CAR T cells, they can cross the placenta. So if you have persistent CAR T cells in treating young girls, when they grow up, if the CAR T cells are still there and they get pregnant, they can cross the placenta. And there have been babies now born after treatment of CAR T cells that they had that they had lower B cell counts because the CAR T cells crossed. Fortunately, those babies, it's been rare, have recovered their B cells, but they are being born with B cell immune deficiencies, and some of those that are hopefully going to stay transient. But I just think it's things that we don't even think about as far as potential side effects of immunotherapies could be there. So they're not necessarily going to completely eliminate toxicity in our patients. We also don't know for a lot of these immunotherapies like blinatumomab, what is the optimal dose, schedule, number of cycles, route, and formulation. So with Blina, the adults studied 4 cycles. In kids, we studied 2 cycles. In infants, they studied 1 cycle. What is the ideal? How many weeks do you give per cycle? There's some data that suggests that we give blinatumomab as a 4-week treatment, but you probably get all the benefit you're going to need after 2 weeks because the T cells get exhausted. There are newer bites coming out the pipe that have longer half-life. Blina has a sub-Q formulation. AstraZeneca and Merck have long-acting bites. Are these going to be more successful, both from an efficacy and a burden standpoint for our patients as well? And these are questions that need to be studied and asked. And then the last thing is, how do we ensure equitable access? Some of these immunotherapies are hard to manufacture. They're very expensive. They are a big burden on our families as far as how they're delivered. In the United States in particular, it's much easier to give Blina to somebody who lives near a big tertiary care center than somebody who lives in a rural part of the country that's 3 or 4 hours away from a hospital if their backpack becomes disconnected. So if we're going to have these new expensive therapies, we have to make sure that every child who might benefit from them has equal access to them, so it doesn't become a world of have and have nots. And I think just to summarize there, where do we go from here? We will start de-intensifying therapy. We will reevaluate how we risk stratify patients. We are going to adapt to the changing landscape at relapse. We are going to reassess who needs a transplant in first remission. We will study the late effects. We will study dose and schedule, and we must ensure equitable access. And with that, I'll thank the members of my lab and people that I've collaborated with both locally and internationally over the years, and the whole COG ALL team, funders, patients, families. Thank you.
Thank you, Dr. David Tichy. Wonderful lecture. Fantastic. It was a true replica of the ASH. Thank you so much.
You're welcome.
And now I request all our discussants to please put the hand raise signs. So we can start the question session with Dr. David Tichy. Yes, first we have Dr. Roshni Pandey, ma'am. Please go ahead.
Good evening, sir. Thank you for that excellent update. My first question is on pH positive ALL. Which of these subsets should go for transplant?
It's a great question as far as pH positive ALL and who would go for. I think one, it depends on what the backbone you're treating with. I think really we're still relying on MRD primarily to say which patients with pH positive ALL need to go to a transplant and those who have a worse response. In the caveat that the time point and the threshold that you use to determine which patients would benefit or should go to transplant varies based on your backbone that you're using to treat the patient. So positive disease, persistent MRD probably still benefits from a transplant. It's just the threshold and the time point of that varies based on your backbone, and it's continually changing. And it'll probably be hopefully less and less patients are going to need a transplant. I think that's one of the things we're learning from adults is they're kind of 2 steps ahead of us now as far as really moving to immunotherapy, TKI-based regimens with less chemotherapy, and they're taking less and less patients to transplant, and they're finding more and more success with that. So it's a rapidly changing field.
In pediatric ALL, which is better, dasatinib or imatinib?
It's a great question. We think it is probably dasatinib because for pH positive ALL, because it crosses the blood-brain barrier better. The only head-to-head comparison was that study from China where they did a randomized study comparing dasatinib versus imatinib in patients with pH positive ALL. They used a different dose of imatinib than traditionally used those. So some people question some of the results of that study because they biased against imatinib. Either way, it looks like dasatinib is a better TKI. And now we're in a world where there's 2nd and 3rd generation TKIs as well, things like pomatinib and asciminib and other ones as well. With pH like ALL with Abel class fusions, dasatinib looks like it's better for most subtypes. However, the PDGFR rearranged, probably imatinib is a better TKI, at least based on preclinical data. We don't know if that'll be true in clinic though.
The next question is CAR T cell just a bridge to transplant? Or, I mean, if we look at, say, pH positive ALL, I mean.
It's a great question. I don't think we have a definitive answer on that one. So easy groups, T-ALL, CAR T cells are a bridge to transplant. Simple. With the CAR Ts we have right now, B-ALL, I don't think there's any consensus in the field. For me, I tend to believe in using the CAR T cells as standalone more now if there is something you could do if the patient relapses after CAR T cells. If it's your last available therapy because it's the only CAR T cell you have access to and you may not be able to get their patient back in remission, the data on using CAR T and transplant are actually pretty compelling. What we do locally at my center is we often intend that CAR T cells will be definitive therapy, but we look for B cell recovery. And if your normal healthy B cells start recovering a few months after getting CAR T cells, we're saying those patients probably need a transplant. But the field is continuing to evolve. What we really need is a randomized study where we randomize patients to get CAR T cells alone, transplant alone, or CAR T cells plus transplant and learn which wins. But that would be very hard to pull off. So I don't think there's definitive consensus on who CAR T cells should be standalone versus who they should be a bridge. And I think different physicians are using it different ways. I wish I could give you a straightforward answer.
Yeah. In early relapses, we know it's a chemo-resistant disease. So how much chemotherapy should we push in the induction? Because we might end up losing lives.
It's a great question. What I like, so 2 things. One, it depends on the group and first relapse. because the part of it is, what is the goal, and do you need response to the first month of therapy to impact your decisions? So for very high risk early relapse, we now in the US are trying to use less intensive inductions with the goal of just a little cytoreduction to get you to blinatumomab, and then use and then take you to transplant. There are some patients who have later relapses that, for example, we don't know who should go to transplant and who should not, and we use end reinduction MRD to help make that decision. So for those patients, we're still giving a VXLD type reinduction to get that MRD answer to it, and then taking those patients to help decide transplant or no transplant. I think that's kind of getting out of favor too. So most groups are moving towards less and less intensive inductions in order to get you to cytoreduce for immunotherapy. That 1821 trial that I mentioned before, that was a trial we ran through COG that right out of the gate, patients relapse, we started treating you with blinatumomab. That was a bust. So without cytoreduction, blinatumomab doesn't work very well. And so you need a cytoreduce first. The next study that we're studying in COG, we're actually asking the question of randomizing patients from a low intensity chemo reinduction. So 3-drug reinduction versus inotuzumab as a reinduction followed by blinatumomab, and we'll have to see how successful that is. So it's, I think, an emerging field, but ultimately I like the UK approach, which is patient relapses, you give them a 3-drug low intensity induction, you repeat a marrow at day 15. If they still have a lot of disease, then you add in an anthracycline and then repeat MRD at the end of that, where the ultimate goal is to get down to less than 5% blasts for blina.
And are seizures an absolute contraindication to continuing blinatumomab?
I don't think they're an absolute contraindication. I think it depends on the type of seizure, the severity of the seizure, but more importantly, because were they on prophylaxis when they had the seizure or not? A patient who got blinatumomab, had a seizure, and wasn't on anything, and then you can put them on Keppra and then re-challenge them sometimes with blina, and actually they do fine. So it's not an absolute contraindication, but it's a relative contraindication.
And in low middle income countries, is there any role of rituximab?
It's a good, I mean, the data from adults using rituximab are actually pretty good. And it's standard now in CD20 positive adults with ALL. So yes, there's definitely a role for that. If you have access to it. And I know that blinatumomab is challenging for a lot of the world for lots of reasons. It's one of the things that makes me excited about some of these newer BiTEs, like the subcutaneous formulation of blinatumomab or the longer acting BiTEs, because they may help reduce some of those issues that'll make it easier to use in lower middle income countries once they can get to be cost effective and cheaper.
Thank you so much, sir.
Thank you, Dr. Roshni Pandey. Dr. Gopinathan, your question.
Good evening, sir. Thanks for that wonderful lecture. A lot of advancements in risk stratification. I think by day and day, it becomes difficult for us to write up in the exams. Previously, it used to be just a single 3-tabular column, but now it's becoming more complex. So I have one case to be discussed. He's an adolescent boy, 18-year-old boy with TALL, non-ETP. It's more of a cortical TALL subtype. This NGS has come to have Notch mutation in the PIS domain. And he also has got a CDKN2A mutation with a VAF of 93%. And this guy has got a pituitary involvement in MRI, but he turns out to be MRD negative at the end of induction as well as consolidation. So my first question is, how will the CDKN2A will have a cooperative effect with Notch mutation? Because isolated Notch is slightly good. And second thing is, should we consider transplant in this patient?
I wouldn't. So a couple of things on that. One, I think the only clear indication for transplant in TLL right now is persistent MRD at the end of 3 months of therapy. Those patients we clearly know need a transplant. Right now, we need some of the genomic data to be validated in independent data sets and independent populations before we make decisions on should a patient get a transplant or not based on genomic lesion whatsoever. So if a patient is MRD negative at day 29, I don't care what their genomics are. Right now, I recommend they just get chemotherapy alone. There is P53, there's no genomic alteration that changes my opinion if you're MRD negative at the end of induction. Until we validate more of these genomic features across other cohorts, because it's not just what are their outcomes, it's do they actually benefit from a transplant. I think one of the things that we often do in ALL is we have this arbitrary, if your cure rate is less than 50 or 60%, you need a transplant. Transplant works pretty well in some subtypes of ALL, but not as well in others based on how important GVL probably is. So the reality is there's probably patients with 80% cure rates that benefit from transplant, and there's other patients with 20% cure rates that probably don't. Hypodiploid ALL is a good example of that. Outcomes in hypodiploid ALL are bad of chemotherapy. They're bad of chemotherapy plus transplant, and they're about the same. So you don't gain that much by doing a transplant in a hypodiploid patient. But again, long and short, for your patient, I would not transplant that patient because based on their MRD response.
Right. And the second question with the patient is if the patient has had some kind of neurotoxicity with vincristine, some jaw pain, some kind of vincristine. So do we also assume that it's a parallel cross toxicity with nelarabine also?
It's a good question. They're probably at a higher risk with nelarabine. It's not an indication not to use nelarabine. And it's just something to watch for that. Yes, nelarabine causes neuropathy, vincristine causes neuropathy, bortezomib causes neuropathy. There's no great data though that if patients get neuropathy of one drug, does that increase your risk of the others? Although the presumption is it was. Nelarabine peripheral neuropathy tends to be reversible, just like vincristine or proteasome inhibitor neuropathy if you don't keep giving the drug. The bigger worry with nelarabine is the central neurotoxicity. And we do know there that peripheral neurotoxicity does not correlate with central neurotoxicity. So having somebody with vincristine peripheral neurotoxicity, I wouldn't worry they're going to increase risk for nelarabine central neurotoxicity, which is what we're really nervous about with nelarabine, because that can be permanent.
Yeah, thank you. And the last question is, right now at this point of time, post-CART, what do we have as maintenance strategies? Because we see some abstracts telling azacitidine, some abstracts telling lenalidomide. So what do we take from that point of view?
I think it's a good question, but it's a complicated one. I don't think there's any universal consensus of which patients should get some kind of maintenance therapy and then what maintenance therapy they should potentially get either after transplant, after CAR T. I think it's an area of active investigation, and a lot of it is the ultimate answer is hopefully on a clinical trial. But like, I mean, it's not just should it be ven and aza, like should it be a targeted therapy? So like if they had KMT2 rearranged disease and you gave them a CAR T cell or transplant, should they get a menin inhibitor at some point as a maintenance strategy? We need more studies right now. I don't think there's anything that we would say is standard of care post CAR T for maintenance, and things that are just being tested on trials.
Thank you. Thank you, sir.
Like at my center where we treat a lot of patients of CAR T, we are not routinely just doing some kind of maintenance. However, if we have a patient that has early B-cell recovery, there's lots of different approaches we're taking towards those. Like sometimes we take them to transplant, sometimes we say, let's try some other chemotherapy or maintenance type therapy, but it's really patient dependent. What have they seen before? What relapse is it? What have they responded to, et cetera, versus a universal approach.
Thank you, Dr. Gopinathan. Thank you, sir. Dr. Rama, your question, please.
Good evening, sir. It was a wonderful lecture, and it was first time for me to listen regarding the CAR T cell for T leukemias. So my question is CD7 CAR T cell. What is the survival benefit over the other available therapies right now? And second question is it can it be applied to the mediastinal TLBLs as well?
So two good questions. One, just to clarify, when you're saying comparing CD7 CAR to other therapies, are you talking about other CAR T cells or other therapies in general? Your second question, so for that one, I would say when you're comparing, as you probably know, that once patients with TLL relapse, they tend not to respond to a lot of stuff. They have highly refractory disease, and it's not just cytotoxics, it's small molecule inhibitors. I think there's a huge role for CAR T cells if you have access to them in the relapse and refractory population, because they tend not to respond to anything. But again, they're a bridge to transplant. What's your second question again? Sorry, I'm now blanking on it. Answer the first.
Mediastinal TLBLs.
Yeah. So it does seem some of the early data, and this is again small n, small centers trials, different CARs, different countries, so do look like that TALL or Marrow disease is more sensitive and better at responding to CAR T cells than some of the lumpy bumpy lymphomatous disease. That said, there's lots of patients with lumpy bumpy T cell lymphoma that are going into full MRD negative, PET negative CRs with CAR T cells. So while they may not be quite as good as they are in T-ALL, they're working just nice. So we've had patients with large mediastinal masses that we gave them CAR T cells, and they went into a remission. So I think it's less likely to be as effective, but it's better than we're seeing with chemo or other like targeted therapies.
Okay, thank you, sir. And compared to other therapies, means CD7, you have told it is already been tried. What about the other CD5 or other CD1A, all those comparatively? Which is the best CAR-T to give right now to improve the survival in the patients?
So I think one of the reasons that CD7 has become so prevalent as far as a lot of the CAR T cells out there is because CD7 is on the vast majority of cases of T-ALL, and T cell blasts tend to care about it, and CD7 doesn't tend to have as much change with time. So unlike B-ALL, where you don't have as much antigen drift, in T-ALL, there's a lot of antigen drift. So if you look at the immunophenotype of a T-ALL case walking in the door versus their immunophenotype at end induction of the persistent MRD versus end consolidation of the persistent MRD versus relapse, the antigens on the cell surface change quite a bit. ETP turns into non-ETP, non-ETP turns into ETP, but CD7 tends to be a stable antigen, so it tends to be prevalent on most cases of T-ALL, and it tends to stay present at relapse and at multiple relapses as well, which is why it's attractive. CD5, in contrast, is negative in about 30 to 40% of T-ALL. So by definition, walking in the door, a third of your cases of T-ALL are going to be CD5 negative and therefore would not benefit from a CD5 CAR. So you have a rare disease and you're trying to think of what should I build a CAR against that's going to be practical to benefit the most patients. You're going to want something that's on most cases. And that's why CD7 has seemed to be the thing leading the field. The data of CD5 CARs are very nice, though. Like some of the data that are coming out, some of the trials are looking very good. It's not positive on a bunch of cases, which is why it's a little behind. I actually think that CD5 CARs ultimately, though, could win because for PTCL, so adults with T-cell lymphoma, which is far more common, they're almost all CD5 positive, and it's the opposite thing where most are CD7 negative, so there's not as much enthusiasm about a CC7 CAR in adults with peripheral T-cell lymphomas as their CD5 CAR, and often what works in a lot of adults tends to be the thing that wins. So I wouldn't be surprised if CD5 CARs go forward. CD1A is similar to CD5. It seems like it's a really good CAR. The data from Spain are really nice. But it's only, in that case, it's only about a third of TLLs are CD1A positive. So 2/3 of cases wouldn't benefit by definition. CD38 is another area that people are looking at CAR T cells because CD38 is expressed on most cases of TLL. And then the holy grail is going to be identifying something that's on T cell leukemia, but not on normal T cells and making a CAR against that.
Okay, that wonderful answer, sir. Thank you so much. So my last question is do you check for the MRD before you start the CAR-T, or you prefer it as an initial treatment and then go ahead?
So usually if we're going to give somebody CAR T cells, we check MRD before that. Like we want to know the disease going into it so we can, one, because it probably does predict likelihood of toxicity. And two, we're learning more and more that patients of bulk disease going into CAR T cells or a large level of disease are less likely to have persistence and then more likely to maybe need a transplant. It's just so things that you can think about in the back of your mind versus those who go into a low level disease. Often the CAR T cell persists better.
Okay, sir. Thank you so much.
Thank you, Dr. Rama. Dr. Shweta Pathak, your question.
Good evening, sir. Thank you for a wonderful your presentation and discussion, sir. Sir, I have few questions. You have said in T-cell ALL there was a use of venetoclax and as I mentioned. So where do you place this veneza in first relapse setting, second relapse setting, or as a bridge to transplant? So because that was very short slide. That is first question. Hello. Yes, sir.
It's all of the above. So I think that with relapse and refractory TLL, there is no real great standard, and quickly you get into what I call kitchen sink mode, which you're just trying different things to get them to transplant. So in newly diagnosed patients, so patients who have persistent disease, so newly diagnosed patients, I treat them at conventional induction. Regardless of response to conventional induction, I follow by a traditional consolidation, ideally with Nelarabine. If at the end of consolidation you have persistent disease, then the goal is to go to transplant, and is how you bridge them. And I typically try one block of chemo even in those patients, because a lot of patients who still have low-level persistent disease after consolidation do go into an MRD negative remission with chemo. For those who have frank disease at the end of consolidation, like 50% blasts, or fail a chemo block, I'm using Ven/Aza as actually my regimen of choice for those patients with refractory disease as the first salvage attempt that I try, and I often do it with daratumumab. So I do Dara, Ven/Aza as kind of a approach to give 3 different agents that they haven't seen before. In relapse disease, first relapse, the best regimen for first relapse is still more traditional 4-drug type induction. So then for those patients, I basically say, when did they relapse and what toxicities do they have? And I either choose UKR3 type reinduction, or I'll use the 07P1 approach, which is a 4-drug induction prednisone based with bortezomib, or I'll use the Delphinus approach, which is a 4-drug induction plus daratumumab. And the rates of second remission are equal on all 3 of those regimens. So I usually base it on the patient and what toxicities they had, etc. After that block, if they get into a good place, I typically use Nectar as consolidation, so Nelarabine VP Cyclo. And then I will use Ven/Aza Dara either to salvage if they still have positive disease, or as a bridge to transplant if they're MRD negative. So I usually don't reinduce with Ven/Aza unless it's multiply relapsed TLL. Unfortunately, with TLL, there's not a one size fits all for me when patients have relapsed or refractory disease. There's a lot of factors that go into it. And then the regimens I choose between are UKR3, Delphinus, 07P1 like Nectar, Ven/Aza Dara are usually the things that I debate in this question of which one do I pick. And if all those fail, then I start just trying stuff like ruxolitinib or palbociclib or other things.
Okay. Thank you, sir. One more question. Usually, the adolescent patients, female, they tend to have many issues with prednisolone, initial prednisolone, the hyperglycemia, those who are obese, especially the hyperglycemia, hypertension. So, somehow, if you finish induction, so is there any role of giving intermittent DEXA during reinduction? Because that time we have to give DEXA for 21 days because we follow PFM 2002 protocol here. So we give DEXA for 21 days. So is there any difference between giving intermittent DEXA in such kids, especially?
So we don't use, for us in COG, in our reinduction, delayed intensification, whatever you want to call it, we don't give 3 weeks of Dex. So it's hard for me to say other than our data looks similar and we don't do that.
Okay. And there's one more question.
But it is hard though, because I do recommend that whatever regimen you're using, you should stick to that regimen and not like pick the COG DI and the BFM equivalent of reinduction. They're protocol 2. Like, you want to be consistent because there are differences. You're risk stratified differently. There's lots of little subtle differences that makes it hard to just pick and choose one block from one group and another. So if you're following BFM therapy, I'd follow BFM therapy.
Okay. The last question, back 1 minute. The last question, when the standard risk B-cell ALL child, when they develop a serious reaction to PEG asparaginase, then we don't use it, and Erwinaze is we don't have access to Erwinaze here. So what do you suggest, sir? Any additional therapy for them? Anything to be added later, or we just skip PEG asparaginase in subsequent reinduction?
So I tend to skip. Like we don't have any data on what you should replace it with, or if it'll be helpful or just cause more toxicity. So my usual bias is, if you can't get more asparaginase, then you keep treating without more asparaginase.
Okay. Okay. Thank you.
Thank you, Dr. Shweta. Dr. Shilpi Saxena, your question.
Thank you, sir, for the wonderful presentation. Sir, I have a case to discuss. A 9-year-old child, she came with history of pancytopenia, grade 2 neutropenia with severe anemia. HB was 3. So on PBS, she had dimorphic anemia, macrocytic plus normocytic. So we gave initially supportive treatment with blood transfusion, folic acid and vitamin B12. There were no blasts on peripheral blood. We went ahead with the bone marrow aspirate and biopsy. So there were 4.3% abnormal bone marrow blast in the bone marrow aspirate. On flow cytometry, there was aberrant expression of CD86, bright CD34, CD2, and bone marrow biopsy revealed more than 20% TDT positive B blast. So we gave a diagnosis of BALL and advised the patient to undergo chemotherapy. But the patient father was a little reluctant. He just took away the patient, and then we sent for molecular studies also. By the time molecular studies came, the patient was almost lost to follow-up. But his so the molecular studies revealed DUX4 IGH fusion. So there was no doubt about the child being BALL, but after 8 weeks of onset, when the child came back, so the counts were improving. He denied any history of taking any kind of chemotherapy or steroid. So our pediatric hemato-oncologist, he was reluctant to give any treatment since the count were improving. So then we just followed up the patient with CBC. So CBC remained normal for a duration of 9 months that we followed the patient. So I just wanted to ask your experience, since you have seen so many patients of BALL. So your experience with spontaneous remission in BALL.
I never seen it. I mean, I know people have reported it, and there are cases of it, but it's extremely rare. So I think you have to meet the diagnostic criteria. So I've definitely seen patients who like walk to the door with like 8 or 10% blasts across all different testing modalities, who don't meet the criteria for BLL, and I have rarely seen or heard of those patients having spontaneous remission. The ones who actually meet the diagnostic criteria with over 20% blast, I've never seen one of those patients have a durable spontaneous remission. I have seen ALL that can kind of wax and wane for a little while. So I would be worried eventually that patient's going to not have better counts. This other thing with BALL is time to diagnosis and time to treatment doesn't actually impact cure rate. So while that patient, I think, is eventually going to need to get treated, you're not losing that much by careful watching and waiting. If you're the risk of that is you could have a bad event happen while you're doing that. But and I think eventually the patient's going to declare they're going to need chemo. But I guess you can see if they're the very extreme rare spontaneous remission, which again I've never seen.
Okay, sir. Thank you so much.
Thank you, Dr. Shilpi. Dr. Vikramjit, sir, do you have another question? Dr. Vikramjit Kanwar, sir.
Yeah, sorry. So I know it's late and everybody's tired. So for Dave, just one, you already touched on this very briefly. You talked about blinatumomab, but I get families wandering in who've seen the NEJM article and assume this is now the standard of care for standard risk ALL. Really, is it fair to tell them it is not yet? It's very promising because most of them can't afford it. They're a little bit linked to control. So what is your feedback on that? It's a tough one. I know you don't have a clear answer.
So, I mean, I think it is one of the hard parts of blinatumomab right now. It's an expensive drug. It's not everybody has access to it, especially in lots of parts of the world. You can cure BALL without blinatumomab, and the cure rates are exceptional without blinatumomab. Blinatumomab seems to make them a little bit better. So if you have access to it, then I would recommend giving it in most patients. If you don't have access to it, then you can still cure patients without it. Most patients without it. So I mean, I don't think there's a black and white, yes, no, right, wrong there. I think you have to treat what you can. Most of the world, I mean, the US is an exception right now about giving Blinna to most patients. Like in Europe, as another example, they have access to it in certain populations, but not all populations, because the European regulatory authorities say they need more data in Europe, in a European population, before they would just make it standard of care based on costs across the board. So do I think in 10 years it'll be the standard across the world? Probably, as it gets cheaper and some of the newer formulations are more readily available. But right now, it is an issue that it's standard if you have access to it.
Great. I mean, time will tell with all things. Thanks, Dave.
Yeah.
Thank you, sir. There are some questions from the audience. First question from Dr. Yimon Tent, 15 years old girl, pH negative, TALL, initial WBC count of more than 400. Would you choose pediatric inspired regimen versus hyper-CVAD?
I would use a pediatric regimen. I think the data are better with those than they are hyper-CVAD.
Perfect. Another question she has is about optimal allogeneic stem cell transplant time for her.
So how old is she? So is she, how much? How old?
15 years female.
I would say she only needs a transplant if she's MRD positive at the end of consolidation. And then if she is MRD positive in a consolidation, you transplant as soon as you can get into a low MRD negative remission. Like it doesn't, I think in TLL there's nice data now from Europe, and we have some in the US too, saying you don't have to be MRD negative in the transplant to still have good outcomes of transplant. You just want to have very low level disease, at least less than 0.1%. But again, the decision on transplant in a 15-year-old is the same as a 5-year-old, is the same as a 25-year-old. It really is based on your MRD at the end of 3 months of therapy. In fact, if anything, in TALL, we have data on this. A 15-year-old does as well as a 5-year-old. 25-year-old does as well as a 3-year-old. It's not BALL, where the older you are, the more risk of relapses. I mean, the older you get, those patients sometimes have more issues of toxicity with asparaginase products, but aside from that, outcomes are equivalent.
Thank you, sir. In those patients who got hyper-CVAD, how about benefit of L-asparginase, which they will not get it?
I mean, there's never been a direct head-to-head comparison of hyper-CVAD versus like a more traditional 4-drug regimen that's used in kids. I don't think it's an answerable question, really. I just think the best data out there for TALL are using a pediatric inspired regimen, but the adult data are good with hyper-CVAD.
Thank you, sir. Another question is from Dr. Raghavendra Rajesh Varkar. You said that when the Blinna is going to be used in future, upfront, many chemo agents will drop out. Which chemo agents will not drop out and why?
So one, we don't know yet. We're going to baby steps because we still haven't had any great data that we can get rid of any chemo and it actually is effective. But I think the things we're going to have the hardest time getting rid of are the drugs that treat the CNS, because Blinna doesn't cross. So for example, interthecals aren't going away anytime soon because you need some interthecal chemotherapy, especially as you're going back and less systemic agents. You're going to make sure you have CNS penetration as well. So I think that interthecals are going to be one place we're still going to have therapy there. I think some of our blocks, like intermaintenance, actually may become more important because they're very good at penetrating the CNS than others. I think we have to be careful as we're getting rid of steroids, because steroids are actually good at prophylaxing the CNS too. So I think time's going to tell, and we'll have to be very careful and meticulous about what we try to get rid of.
Thank you, sir. Another question from Dr. Vasi. How do you proceed with steroid dose at pre-phase and induction? Do you go with prednisolone 40 mg per meter square first for pre-phase for 7 days and then jump to 60 mg per meter square for the next 29 days? And then do you abruptly hold the prednisolone without tapering? Is that possible?
So I usually am a believer of follow the regimen that you're using. So in COG, we don't do prednisone prefaces in any group, except for infants. So we don't do it. In BFM, they do, as you said, use their traditional prednisone prephase, which is usually like 40 per meter squared, and then they upramp it. I would follow the protocol you're using, and follow it accordingly, and not like try to change the taper or do it a different way. I would, if you're using BFM regimen, I'd follow the BFM protocols.
Thank you, sir. Another question from Dr. Anil Kumar Yadav. What is the role of deletion of TLX3 allele, which is located on chromosome number 5, as well as deletion 5q in ETP ALL?
I mean, they're involved in the biology of it. I don't think we have any data right now suggesting that impacts outcomes at all. We do know that the TXL3 subtype of TALL, ETP ALL, where it's rearranged, those patients actually do pretty well. I don't know the deletions off the top of my head as far as if they do worse or better or similar. So I guess the answer is it's involved in the biology, but we don't know the clinical relevance of it.
Thank you, sir. Another question from Dr. Shailesh. pH like ALL, 44 years old male, B cell, pH like ALL, CNS 1, karyotype normal, NGS shows BCR JAK2 fusion. How would you treat this condition? Do you suggest to add rituximab upfront?
I wouldn't, based on the data we have, unless that with a fusion, if the patient was high level disease, MRD positive at the end of induction, I would think about adding in a JAK-STAT inhibitor, but I'm usually pretty resistant to do it unless they have high level MRD, because we need data.
And if you're using ruxolitinib, then what is the dose, duration, and how would you incorporate it in hyper-CVET protocol?
So I would say that I would move them to a protocol that it's been studied safely. So for us, we've used ruxolitinib in a BFM type consolidation, so we know how to dose it there. I don't know the dose off the top of my head, but it kind of goes back to published regimen that's used it safely and effectively. Follow that.
Thank you, sir. Another question from Dr. Anil Kumar Yadav. In the present era, many genome profile is categorized as pH-like. So translocation 1221, like ETP, ALL, etc. So is it maybe in the future, all these entities will be bifurcated because of all these novel therapies coming up?
I mean, I think that, like, if you look at the data that came out of St. Jude, most subtypes of ALL are mutually exclusive. So, I mean, pH-like is an expression profile, but there are other groups that have the same expression profile, but if they have the somatic alteration, they don't get called that. So ETB6 Trunks1, they can have a gene expression profile that looks like pH-like, but if they have the fusion, they don't have pH-like ALL. So pH-like ALL by definition is having a specific gene expression profile and not having some of those other signatures. So I think they all are mutually exclusive things. I think that's what you're asking, but I'm not 100% sure what your complete question is.
Yeah, because the same question, because whether this will be bifurcated in future, I think yes. So
Yeah,
another question. Yeah.
I think it also depends on the trial and the questions that you're asking as far as how you treat different molecular lesions.
True, sir. Thank you, sir. Another question from Dr. Kannan. If MRD remains positive, will CAR-T be the better option than the transplant?
What setting? So MRD remains positive in TALL after consolidation, or BAL? Like, what's if it's TLL, CAR T-cell options aren't that much. In BALL, we don't know the answer to that right now. For in-consolidation MRDs, transplant is considered standard of care. However, there are groups that are using CAR T-cells in this setting. There's no data, so we just don't know. There was the ALL 1721 clinical trial that was run in the US and Europe that took patients that were persistent MRD positivity at the end of consolidation and treated them with CAR T cells. But the results of that trial are not released yet.
Thank you, sir. Another question from Dr. Shailesh. 23 years male, B-ALL, normal karyotype, CNS1, fusion is EBF1 PDGFRB, post-transplant. do you suggest to go for dasatinib maintenance post-transplant? And if yes, how long?
Yeah, it's as far as a routine practice, for PH like ALL, we're not using TKIs at my center as far as the routine. So we would only do it if they had resurgence of MRD post transplant, then we would do it. But if it's PDGFR beta, I'd probably use imatinib actually.
And maintenance, how long?
Yeah, I don't think there's great data on that, so I can't even make a good recommendation.
Thank you, sir. Another question, last question from Dr. Shyam. PH positive ALL, first relapse, underwent allogeneic stem cell transplant. Second relapse, underwent CAR T cell therapy. Third relapse, which one would be the best? 1 year of CAR T cell therapy, and the patient is currently on TKI plus chemo in remission. What will be the next line of treatment? Bina repeat or CAR or second allo stem cell transplant?
It's a tough answer because I guess what kind of transplant did the patient have the first time? Was it TBI based? What was their MRD going into transplant? What was MRD coming out of transplant? How soon did they relapse after transplant? What are their donor options? Like, I think with mostly relapsed ALL, there's no like one size fits all answer. So I would need a lot more information to make a definitive recommendation. Feel free to email if you want the case, and I can say what we would consider doing. But I think it's hard to say that second relapse after a first transplant is standard to do CAR versus to do CAR plus transplant versus transplant. It really depends on the patient.
Yeah, because this patient actually that is a relapse within 1 year of CAR T cell therapy, and patient is
Gotcha.
currently on TKI plus chemo in remission. I think difficult to answer. Yeah, thank you so much, sir. Dr. Shilpi?
Sir, thank you, sir. Your comments on role of CSF flow cytometry at diagnosis and at follow-up in deciding CNS involvement in cases of BALL.
So in Europe, they're using it pretty standard. In the US, we don't use it for the most part because we can't manage to make it happen. Like, so most labs in the US can't do CSF flow accurately or reliably. So it's like in my center, we can't even order it.
Yes, sir. Thank you.
Thank you, Dr. Shilpi. Dr. Roshni, ma'am, you have another question?
How do you treat biphenotypic leukemia, like ALL or with an AML induction?
So we usually what we do and what I do is we usually start with ALL induction, and then based on response, either continue on ALL type therapy or switch to AML therapy.
Thank you.
Mainly because like the data are that most patients with biphenotypic leukemia respond to ALL therapy. There are some that don't. We can't predict who does and who doesn't at diagnosis, and ALL therapy is less toxic. So I think a lot of groups favor ALL type therapy as the first choice because if you can get away with ALL treatment, that's much easier than doing AML treatment.
And in pediatrics, do all of them need transplant? Because I mean, some anecdotal patients have done well even in my practice. Just treated by ALL.
I don't think all MPAL patients need transplant. I think a subset do, but it really depends on response to the first. I think anyone who still has high level MRD at the end of a reinduction needs a transplant. Anyone who fails ALL therapy needs a transplant, but it's not all patients with MPAL need a transplant.
Thank you.
Thank you, ma'am. Another question which has just popped up. Dr. Anil Kumar Yadav has, is it possible that two high-risk genetic markers like BCR-ABL and KMT2A could be positive together?
It is extremely unusual. They're usually considered mutually exclusive for those two. There are some high-risk lesions that actually you can have in the same patient, like IAMP21 can sometimes be in PH and CRLF2 rearranged, but BCR-ABL and KMT2A are typically mutually exclusive unless you have two leukemias.
Thank you, sir. I've finished all the questions from the chat box. Any discussion over here? Has any other question? Well, so there are no more raising signs, and I finished all the questions on chat box, sir. Thank you so much for the fantastic and lovely lecture, and what a wonderful discussion we had with you, sir. Thank you so much.
Thank you so much. Thank you so much. Take care. Thanks for having me.
Thank you, sir. I hand over to Dr. Suyash Bharat for the final vote of thanks.
Oh, thank you so much, Dr. Dev sir, for excellent session. You handled all the queries. In fact, it was a great learning for all of us. Thank you. With this, I'd like to thank MHG, Dr. Amit Khurana sir, Dr. M.B. Agrawal sir for letting us be a part of this session. I would also like to thank Perfect Square for manning the session altogether. Thank you, sir. Thank you, everyone.
Thanks a lot.
Thank you so much. Take care.